A cell membrane permits some materials to pass through while keeping other materials out. Such a membrane is called selective permeability membrane. Under normal conditions, water constantly passes in and out of this membrane. This diffusion of water through selectively permeable membrane is called osmosis. Like other substances, water diffuses from a region of higher concentration to a region of lower concentration. When the transfer of water molecules in and out of the cell reaches the same rate, a state of equilibrium is reached. If the concentration of water molecules is greater outside of a cell, then the solution is hypotonic to the cell. Water will move into the cell by osmosis. The pressure against the inside of the cell membrane will steadily increase. If the pressure becomes great enough, the cell membrane will burst. A solution is isotonic to the inside of the cell when there is the same concentration of water molecules on the inside and outside of the cell membrane. To maintain equilibrium, water molecules move into and out of the cell at the same rate. Suppose a living cell is placed in a solution that has a higher salt concentration than the cell has. Such a solution is said to be hypertonic to the cell because there are more salt ions and fewer water molecules per unit volume outside the cell than inside. Water will move from the region of higher water concentration, that is inside the cell, to the region of lower water concentration, that is outside the cell. The concentration, therefore, would change. The selective permeability membrane does not allow salt ions to pass into the cell. The cell shrinks as the cell loses water. So we're going to show you here in this lab with using red blood cells, Euladiya cells, and then also our paramecium to show you the different states, hypotonic, isotonic, and hypertonic, as I just described. So let's take the red blood cell. We're going to put it into a hypotonic solution. And as you can see, it expands very large. Let's move it to an isotonic solution. It's a normal state. It kind of looks like a lifesaver upside down, so see how we can bring that in. Let us move it to a hypertonic, and it shrivels up. The water's going out. Please note the arrows. Let's do that again. Arrows showing inward. Arrows showing in and out, right? And then arrows going out. Euladiya cells now. Again, look at the cells, the arrows. Paramecium next. Put in isotonic first. Let's put in hypotonic. And then hypertonic. Again, if you want to see these again, just rewind the video and check it out. Please answer the questions that are given to you.